Welcome to the SBGrid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SBGrid.
So Yi-Wei is joining us from the Perelman School of Medicine at the University of Pennsylvania. So Yi-Wei, go ahead and share your screen. Great. And so,
okay.
Yeah, it looks good. So Yi-Wei's title today is Cryo-ET Reveals Structures and Potential Regulatory Mechanisms of the Rhoptry Secretory System in Apicomplexan Parasites. Sorry, I sort of stumbled through that. That's a lot of, but it sounds super interesting. I'm looking forward to your talk. Whenever you're ready, go ahead.
Okay, thank you so much. And thank you so much for the invitation. So can you see my cursor?
Yes.
Awesome. Okay. Yeah. Hi, everybody. Thank you for tuning in. So I'm Yi-Wei from UPenn. So my lab is specialized in using cryo-electron tomography, Cryo-ET, to look into frozen cells in 3 dimensions so we can understand how big molecular machines structurally conduct their function in situ. So for example, if we use Cryo-ET to look into a bacterial cell, and the resolution will allow us to see, for example, individual ribosomes inside the cell. In some cases, we can even see different subunits of those ribosomes. But very importantly is that in this type of sample, not only protein structures are being well preserved, but also the membranes are well preserved. As a result, Cryo-ET is a very useful tool to study structure and function of big molecular machines embedded in a membrane, okay, in a native membrane, which could be challenging to be purified out for other methods like single particle cryo-EM to study in vitro. So recently, we have been using this advantage to try to understand the efficient mechanism of Apicomplexan parasites. So this is a big family of single-cell eukaryotic parasites. They include several well-known pathogens such as Plasmodium, Toxoplasma, and Cryptosporidium. All of them cause serious human diseases such as malaria, which is caused about 500,000 deaths worldwide each year. Okay, so and all of them have pretty complicated life cycle, involve other type of hosts, such as mosquito and different animals. But the common thing is that when they come into our body, they will use a defined tip of their cell to invade our cell and form a vacuole inside and to propagate. And then in the end, lyse our cell and then come out. And this repeat lytic cycle of infection will cause the symptom. And for example, this is a movie showing a Toxoplasma cell now infecting a big mammalian cell in the culture on the background. So you can see in the movie, this cell running around and then sneak into the host at the location pointed by the arrow, like opening a portal and then go in. And once they grow into a certain number, they'll be ready and then lyse the host cell and then come out and ready to invade the neighboring next victim host cell. So at this point, we are very interested in learning more about the detailed mechanism at this invasion stage, the host invasion stage. Okay, and that is known that for this invasion to happen, one special organelles inside this parasite called rhoptry need to secrete its content. Okay, it's known that some of the rhoptry content, once it's secreted, will come to the surface of the host membrane. And then those proteins will then interact with some other proteins on the surface of the parasite and then form a ring-like tight junction. And this junction then in turn interact with some ectomyosin-based motor inside the parasite. The parasite will use the motor to move the tight junction throughout the entire body from the tip to the basal end, like wearing a clothes. Okay, in the end, will produce a vacuole by the host membrane and then engulf the parasite in, and then become the infection niche for the parasite to grow in. Okay, and some of other rhoptry protein is also known to secrete onto the membrane and then modulate the property of the vacuole to establish the niche. And some of the content even go through the membrane and go deeper into the host, modify the biology of the host cell, further establish the infection. Okay. And traditional EM have imaged this area of the parasite and then revealed the ultrastructure of the rhoptry to this level. And people saw that the rhoptry is this elongated structure, contains a thin neck pointing toward the tip of the parasite cell, and also contains a larger bulb region toward the center of the parasite cell. By immunogold labeling of different components, people found that some of the rhoptry proteins only localized in the bulb region and some other localized only in the neck region. So there is a clear segregation of rhoptry proteins in the lumen. But the resolution of traditional cryo-EM was not able to reveal how this protein segregation was taking place, and also how the structure, the defined structure of the rhoptry is being maintained. And also very importantly, we don't know yet how the rhoptry is communicating with the outside world for its secretion. And moreover, there is a very interesting difference in terms of rhoptry numbers in each cell among different organisms. For example, Cryptosporidium only has one rhoptry in each cell, Plasmodium has 2, and Toxoplasma sometimes can have more than 10 rhoptries per cell. So how different organisms coordinate and handle different number of rhoptries in each cell is also poorly understood. So we set out to study all those questions by applying cryo-ET on this apical region of this type of parasites. So two amazing postdocs came together. One is Shaman, a cryo-ET expert from my lab, and Amandine, a parasitology expert from Boris Stripan's lab in our school. So together they prepared some Cryptosporidium sporozoite cells, which is the invasion stage, life stage of this type of parasite. So they froze those parasites on cryo-EM grids. Those are individual parasite cells. And then they can locate the apical region of those parasites, and then use higher mag to take projection images while turning the sample degree by degree and take this TIL series. And then use that, they can reconstruct the tomogram of this area of the parasite. So slice by slice, while going through these tomograms, right away, we can appreciate the high structural complexity of this part of the parasite cells. So through three-dimensional segmentation, we can see exciting features everywhere, including those proteinaceous rings toward the tip of the parasite cell, and those arcs. Those are filamentous structures formed by turbulence named conoid. And this purple ring is another proteinaceous ring called apical ring, serves like a organization center of microtubules, which is those called subpellicular microtubules, organized in a helical fashion, going into the cell body underneath their membrane. So together, all those structures form a cage-like enclosing, okay, contains a lot of important organelles and pointing them toward the apical end of the parasite. So among those important vacuoles inside this cage, there's one which is our interest, the rhoptry secretion system, which is this orange membranous organelle. So first, we try to look at the tip of the rhoptry and see how the rhoptry is interacting with the membrane of the parasite. And surprisingly, we never saw the rhoptry directly interact with the membrane, but instead, we always see some densities, we call tip densities, at the tip of the rhoptry. And then we always see a big membrane vesicle in front of the rhoptry tip. And then the vesicle is somehow docked by some proteinaceous density on the plasma membrane. So this is the real tomography slice through this rhoptry secretion system. So this is the part of the rhoptry. You can see the membrane of the rhoptry. And then this is a big membrane vesicle, which is named apical vesicle, which actually have been observed by some other traditional EM early on, but haven't been reported have any association with rhoptries. And here we show this as this is actually at the tip of the rhoptry. And this is the proteinaceous density somehow docking the AV on the plasma membrane. Okay. And in a closer view, we can see very extensive interactions between the protein density to the AV and also to the plasma membrane. Okay. What is also fun is that if we cut through the tomogram from this angle and rotated the sample 90 degree, density 90 degree, we can see this flower-like structure have 8 clear petals. Okay. So, but because of the thickness of this part of the parasite, this is about like more than 500 nanometer thick already, it's approaching to the thickness limit of Cryo-ET. So because of that, the resolution of our tomogram is not the best. Okay, but we were able to collect a lot of tomograms of the same type of parasite. And then in each tomogram, we were able to find the AV region and crop out the subtomogram area and then align them together and generate a subtomogram average to boost the contrast. And we were able to resolve resolve the structure like this. This is the plasma membrane. This is the AV. And those are protein densities linking the AV to the plasma membrane. So in 3D, we were able to segment out different components in this structure and shows that this is actually an amazingly intricate and multi-component machinery anchoring the AV. So which has different anchors shown lighter blue and darker blue here, anchoring the AV outside and inside. Okay, and those linkers, one after another, link it all the way to the membrane anchors shown orange here. Okay, inside the AV, we also see an amazing structure, look like a barrel. And we name it a posterior central channel because we think they might form a channel-like function. And there's another channel called anterior central channel sandwiched between the AV and the plasma membrane. And in front of the posterior channel, there's this we call radiating spoke structure, likely serving as a gate in front of a putative membrane channel inside the membrane. Okay. And because of the location and structure and some additional information I will show later in my talk, we decided to name this entire assembly rhoptry secretory apparatus, RSA. Okay. And if we rotate the sample and look from the surface on the cell, we can see a rosette-like arrangement of densities. In the center, there is a channel-like density in the membrane surrounded by 8 anchors, like petals. Sorry. So those are likely the first line of interaction between the rhoptry secretion system and their host. Okay. And additionally, we also look into the lumen of the rhoptry organelle. Inside our tomogram, based on the morphology of the rhoptry, we were able to clearly define where is the bulb, the larger bulb, and where is the rhoptry neck based on the defined thickness. And there is another slightly larger empty place called tip. And interestingly, when we look into the tomogram, in the central cross-section of this neck region, we always see this continuous dark lining the membrane of the neck region. In 3D, there are actually helical filaments lining the membrane. And we found out the filaments are actually somewhat dynamic because sometimes we can clearly trace individual filament and see they are 2-started helix, like shown here, blue and orange. But in some location, they are 1-started helix, so they are kind of flexible. Okay. And also, if we look at here, the transition region from the neck to the bulb, we can see when they transition into the bulb and larger bulb area, the filament will disappear, suggesting that those filaments might give rise of the defined width of the neck region, like a scaffold. And also interestingly is that we did not see any physical barrier between the neck and the bulb region for protein segregation inside the lumen. So likely the filament also participated in protein segregation in the lumen by preferentially binding to some proteins and exclude some others. Okay. So together, these images give us better understanding about the structure of the rhoptry secretion system in Cryptosporidium. Okay, first we found this helical filament likely shaping the neck and also involved in the protein segregation in the lumen. But more importantly is that we discover a cool big molecular machine at the tip of a rhoptry, likely participated in controlling or regulating the secretion of the content. Okay, and that involved a big membrane protein complex and also a big membrane vesicle. Okay. And this is the cross-section of this part of the model. And this is the plasma membrane of the parasite, and this is the membrane of the AV, and this is the membrane of the rhoptry. And this provides us the first structural framework to start to thinking about how this machine might regulate the secretion of rhoptry contents. So very likely upon the decision of secretion, maybe there are some big conformational change will take place and then allow these two membranes to come together and then fuse and then allow a channel to form to allow the content inside the rhoptry to secrete out. Okay, this is one of the most possible mechanisms for this big molecular machine. But that is also very interesting because if the goal is only to fuse two opposing membranes, say the plasma membrane and the rhoptry membrane for the content of rhoptry to go through, why don't the cell only use some small proteins like snares? They are much more efficient, much smaller for this job. Why the cell need to further involve another membrane vesicle in between, and even with some channel-like structure inside the vesicle? Maybe it's because this procedure is only half of the story, because the content actually need to go through another layer of membrane, which is the host membrane, and to go to the final destination. So maybe the involvement of this membrane and additional structural features actually needed for breaching through the host membrane. So this is actually our future active research direction. Now we're trying to understand by imaging them with the context of host invasion. But back to the beginning, I mentioned that there are interesting differences in terms of rhoptry numbers per cell among different organisms. Cryptosporidium only has one rhoptry, but Toxo sometimes can have more than 10 rhoptries. So we try to understand whether there are some interesting difference in terms of regulation inside the cell. So we collaborate with Maurice Lebrun's lab in France and image some Toxoplasma cells by Cryo-ET. And this is the tomogram of Toxo, and slice by slice, we're going through the tomogram, and right away we can also appreciate the structural complexity of this apical region of Toxoplasma cells. In 3D, we were able to segment out different features inside these tomograms. We see similar things, the two rings at the tip of the cell, and this tubulin formed conoid structures like a cage, apical polar ring, and organizing all of subfollicular microtubules. Entire cage is organizing a lot of important organelles, pointing them toward apical tip. Here, we also focus on the rhoptries inside this area. And indeed, we also saw some tip densities on the rhoptry, and then we also saw a big membrane vesicle in front of the rhoptry docked by the proteinaceous density on the plasma membrane. But in addition, we always see two rhoptries docked to one AV in each tomogram, compared to Crypto. Crypto only has one rhoptry. After we collected many tomograms of Toxo, conducted subtomogram average of this part, we were also able to resolve higher resolution details of those protein structures docking the apical vesicle on the plasma membrane. Okay. And 3D segmentation of those structures revealed a similar organization like what we saw in Crypto, including different anchors, the lighter blue, darker blue anchors anchoring the apical vesicle all the way to the transmembrane anchors shown orange here in the membrane. However, what's different is that here in Toxo, we did not see anything inside the apical vesicle after subtomogram averaging. This basically is empty vesicle. But other than that, we also saw the posterior channel sandwiched between the apical vesicle and the plasma membrane. And we also saw this radiating spoke structure in front of the protetic membrane channel on the membrane. Okay, so this observation suggests that this type of Apicomplexan parasite family have a conserved mechanism, structural mechanism to cover the secretion of rhoptry secretion organelle. Okay. And we also look into the lumen of the rhoptry in Toxoplasma cells. So we can also clearly see the neck region inside the tomogram. And furthermore, we were able to see different subregions of the neck. They have slightly different geometry and thickness of their lumen. First is this anterior neck, which has slightly thinner dimension. And another is this posterior neck, which has a slightly larger diameter. And this is the tomogram of this part of neck. And these are the bottom slice, middle slice, and top slice of this neck region. So we can see clear striation at bottom and top slice with different angle. And in the middle slice, we can see those dots lining the membrane. So this suggests there indeed is, again, a helical filament lining the membrane. And at this region, we can also see those filaments at the bottom and top slice, and suggesting there's also a helical filament. But interestingly, this one is a right-handed helix, and this one is a left-handed helix. And this one is looser in packing, and this one is very tight in packing. And because of this different helix and handedness, we think that they are formed by different proteins. Okay. And furthermore, because this part of the neck is very rigid, so we were able to go into our tomograms and crop out overlapping boxes along this region, and then superimpose all those boxes by different type of searching, clone sampling, rotation, and translation along their axis, and generate a subtomogram average to boost the resolution from this to that. And what we learned here is that, in fact, each striation we see here composed of two parallel lines. So in fact, this is a double-stranded helix, and each strand has a clear anchor anchoring into the membrane. Again, support that this filament provides a structural is a factor forming this neck region, define the shape. Okay. And furthermore, in our tomogram, in addition to the two rhoptries, we always see them docked to the AV at the tip of the cell. We can sometimes also see other rhoptries down there, deeper in the cell. When we look at the tip of those undocked rhoptries, they actually look very similar, almost identical to the tip of those docked rhoptries. They also have those tip densities and empty space of the tip lumen, and also those filaments shaping them into the posterior neck with defined width. So we think those tips seems as ready as those docked rhoptries, they just didn't get their turn for secretion or get ready for secretion. Okay. And also we always see two microtubules pointing directly to the AV. They are colored purple here. And on those two microtubules, one of them always have a chain of membrane vesicles lining up, associating with them. And closer look on those vesicles, we can always see those densities linking those vesicles onto the microtubule. And so that's the reason we start to call those vesicles microtubule associated vesicles, or MVs. Okay. And interestingly, in some other tomograms, we can actually see those unducted rhoptry tip. Instead of interacting with the AV, they can somehow seems to associate with those MVs. And the AV and MV have very similar shape and size, even a similar layer of coatings on the surface. So all those observations allow us to hypothesize, oh, maybe those MVs are precursors of AV. For example, one AV might only be able to coordinate the secretion of two rhoptries. And once the two rhoptries are secreted, somehow the AV might be consumed. So then those MVs might become the new AV to coordinate the secretion of another set of two rhoptries. Okay. So we started to look back to our tomograms to find additional evidence support this hypothesis. So these are 6 examples of many tomograms we took. So basically, in all of the tomograms, we were able to find these microtubules pointing toward AV, and each tomogram might have different lengths in terms of the microtubule length, and also the number of MVs on those microtubules. It could be 3, sometimes could be 5 or more. But they all pose a similar behavior, which is tightly packed toward the tip of the microtubule. Okay. Only the space behind the last MV is pretty variable among different tomograms, as well as the distance between the last MV to the second last MV is also a little bit variable among different tomograms. And some of them even look like this one is ready to go onto the microtubule. So this suggests that those are actually might be loading onto microtubules one-on-one and moving forward and then pack in front, like getting in line. So this type of packing might be guided by molecular motors, moving those vesicles as cargoes on this microtubule track, or be carried out by the dynamics by the microtubule itself. Because in our tomograms, in addition to seeing these microtubules and also those MVs, we also saw these filamentous structures spreading out from the microtubules. Those looks very similar to the intermediates of microtubule dynamic structures. So maybe the microtubule itself is undergoing some assembly and disassembly activities for moving the MVs forward. But this is a way for further analysis to confirm. Okay, but all these observations suggest that this system involving microtubules and vesicles are pretty dynamic and maybe involved in important cellular functions. Likely is reloading of the AV for sequential secretion of multiple rhoptries in this type of organism. So together, these observations we got in Toxoplasma gondii tomograms allow us to reveal a conserved molecular machine likely guiding the secretion of rhoptries. But in addition, this organism likely evolved with additional add-ons for sequential secretion of multiple rhoptries. And this observation is actually conserved to other EM studies of other type of Apicomplexan parasites. People actually reported that among many other papers, when people use EM to look at Apicomplexan parasites known to compose of more than 2 rhoptries in one cell, people have reported the observation of these microtubules or this chain of vesicles or both. But for those Apicomplexan members contains only 2 or 1 rhoptries in the cell. For example, Cryptosporidium or plasma or Plasmodium, they don't have this type of system observed in the cell. Again, suppose this might be related to reloading of AV. Okay. So this study so far has provided all those cool observations, really allow us to start understanding how the rhoptry secretion system can take place. But one key thing to have to further understand how this thing works is to identify what proteins are involved in this type of big machines, so we can conduct mutagenesis analysis and with imaging analysis. And to that end, we were very lucky because when we are doing Cryo-ET on Toxoplasma cells, our collaborator, Maurice Lebrun in France, they were using another type of EM imaging to try to understand this system. They were using freeze fracture replica EM. So basically, they froze toxo cells in a big chunk of ice and use a big cold knife to scrape away a big chunk of ice. And where the ice break apart is normally the weakest point in the cell. And very often it's in between the lipid bilayers. Okay, so as a result, the transmembrane proteins will be exposed to the surface, and then they can ram down the platinum with an angle to create a shadowing effect, and then coat the entire surface by carbon to create a support, and they use organic solvent to wash away biological materials. And in the end, they can get a replica of those transmembrane proteins and image this replica by TEM. And this is the image they got of the tip of the Toxoplasma cell. They can see this rosette structure, just like what we saw of the surface view of the RSA by Cryo-ET. But this image reminds them what has been reported before in cilia. Okay, so if we look at the final the tree of life, Apicomplexan is involved in a big phylum called Aviolata. Okay, inside this big phylum, there's ciliates. Okay, and cilia is a single cell organism. They like to swim in solution by a lot of cilia. Okay, and normally they just chill in solution, but upon some specific stimulation, they will shoot out all those crystalline needles, likely for the function of self-defense. Okay, and those needles are called trichocysts. Traditional EM of thin section of cilia have revealed that those are crystalline needles can be secreted from activities of the core and out from the cell surface. Okay, and over years of study, people already know a lot of things about the protein composition of the system. And also people have tried replica EM on the surface of the cilia. And people have found that the surface of the cilia actually covered by this type of rosette structure. Looks just identical to what Maurice found by using replica EM on the Apicomplexan parasite tip. So this provides a connection between Apicomplexan and cilia. So because in cilia, people already found proteins involved in the formation of this rosette. Those are a set of genes called non-discharge genes in cilia. Okay, so through collaboration with Aaron in University of Chicago, who is an expert in cilia field, Maurice and Aaron, they were able to do an in-depth genetic analysis comparison between cilia and Apicomplexan. And indeed, they were able to find those genes in Apicomplexan. And then they found that if they do conditional knockout of those ND proteins, one is ND9 and now it's ND6 in Toxoplasma gondii cells. In fact, the invasion can be largely disrupted. by immunofluorescence labeling of the ND6 protein in Toxoplasma gondii cell. This is the in one toxo cells. They indeed can identify the protein is localized at the tip of the parasite, and the RNG1 is another protein known to be localized around the tip of the parasite. So they identify the location and function of the ND protein really suggests that it could be involved in rhoptry secretion. And furthermore, by applying Cryo-ET on the ND protein knockdown, the ND9 protein knockdown, we show that in wild type, this is the plasma membrane, this is AV. The AV can be properly docked by the RSA to the membrane. The side view and the top view shows the beautiful Rosette-like structure. But in the mutant, the docking of the AV start to be disrupted, become farther away, and then the Rosette structure indeed got affected. It's no longer a beautiful structure. Again, support indeed the ND protein is involved in the rhoptry secretion system. So together, all those results now allow us to make the link that within this big Aviolata phylum, okay, there are a set of protein forming a Rosette-like assembly in the membrane. In the ciliate family, they are organizing or coordinating or regulating the secretion of the trichosis organelle for shooting out the guns, crystalline guns for self-defense. But somehow along evolution, this molecular machine is adapted by the Apicomplexan members and then added with another membrane vesicle, allow them to secrete the content across another layer of membrane into the host for them to conduct host invasion. So which is a very cool connection we have found. Okay. But this very importantly is that this study now allows us to start learning what kind of protein is involved in this system. So Malis were able to start to do co-immunoprecipitation or proximity cross-linking mass spec to really start to identify players, different proteins really in the vicinity of this region and then involved in the rhoptry secretion. So this is a very recent review published by Maurice Lebrun's lab. This is the tip of the parasite and this is the host. So slowly we can start to assign potential ND proteins at different locations of this secretion system. And now we are actively trying to imaging different mutants individually and try to study the effect of the structure and function and try to dissect the molecular machine of the rhoptry secretion system in situ by Cryo-ET. Okay, so this has been fun. Okay. So with that, so I would like to thank all the people involved in these studies. So of course, all those work will not be possible without strong collaboration with Boris Stripen's lab in our school and Maurice Lebrun's lab in France. And Amandine is a postdoc being trained by these two labs and also my groups. And people with name and labeled are involved in this study. Shawan is a Cryo-ET guru in our lab. And Billy is an undergrad student established the high throughput data analysis pipeline for all of our Cryo-ET study in the lab. And Matt and Liam for their extreme useful data analysis of the study. And thanks again to SBGrid for the invitation and opportunity for me to speak here. And these are the funding source to support our study in the lab. And thank you so much for your attention.
Well, thank you very much. It's fantastic. Those are fantastic, beautiful molecular machines. They're well illustrated by your great graphics, I have to say. They look really looks fantastic. Questions? You can send questions by chat. You can raise your hand. There's a reaction button in Zoom. You can raise your hand. Click the raise hand, and it will raise you to the top. You'll get a little raise hand icon, and then we can you can unmute yourself and ask your question. So anybody questions? I mean, I had one, but it's not so much a question as much as sort of what's your idea about why you need this apparent complexity, this gigantic sort of warhead with dozens of components and helices, and to be able to push your way to me anyway, is a thin membrane that organism should be able to make its way across pretty easily, right? I think maybe it wants to preserve its new home. Maybe it wants to do something before it can get through. What do you think?
Yes, definitely. So one thing is definitely the regulation. They want to fire at the time they want it to fire. So definitely it does not want to random fire into like a void and then just doesn't function properly. So regulation definitely is a key. So maybe, and also because there are multiple layers of barriers they need to go through. So this kind of warhead definitely maybe is designed to delivering things through different layers of membranes at different stages of secretion, because they need to first put things on the surface of the host and then put something over the host membrane into the cell. So there are different stages of secretion. And also the filaments inside the lumen, like I said, one possibility might be involved in organizing contents in the lumen before and even during secretion. Another might be force generation, how they push those contents through those barriers into different compartments they want to put things to. Okay, so those things are still at the stage of hypothesizing, but I think can be answered if we can imaging the active invasion events by, say, combining cryofilm milling and imaging the parasites actively invading their host. So we might be able to provide the next level of understanding of this system structurally.
Yeah, that would be fantastic, right? So a mechanistic sort of picture would be fantastic. Pete Meyer has a question from the group. Pete, go ahead.
Thanks. That was a really cool talk, and I've got I guess kind of a two-part question about the AV vesicles. So I guess like and this is from the tomograms, this pretty clearly highlights that this seems to be kind of a central element. Do we know what's in them, and what is that vesicle doing, or is that one of the other things that still needs to be sorted out?
Yeah, that is the biggest puzzle. We don't know. So what we have higher confidence is that it does not seem to be generated from the rhoptry. Yeah, they seem to be generated separately from the rhoptry, because some of the tomograms we can clearly see there is already a AV docked at the tip, but there's no rhoptries around. So feels like they could be like generated first go there and then some rhoptries go there. Because so it's not first have a rhoptry binding and then somehow pinch away a small ball in situ. Yeah. So as a result, as it makes sense to us to propose it has different content within versus the contents in rhoptry. So maybe the fusion of the AV to the rhoptry somehow can catalyze some specific chemistry event, somehow further trigger the secretion or opening up of the channel. So yeah, we don't know. Yeah, so far we only have those information.
I mean, the part you already know, it sounds like, is does the AV come before the rhoptry or after? And it's, you already know that one. But
yeah.
And I guess the follow-up is for the species that have multiple rhoptries. For Toxoplasma, it's got two rhoptries in one vesicle. Is it known if it's always going to be a single AV, no matter how many you've got? Like if you've got 12, or is that something that varies, or still an open question?
Yeah, so still an open question, but based on our current tomograms, we could not find other rhoptry, other RSAs on the apical end. We think there's only one RSA and one AV at a given time. And then on that single RSA and AV, we always only see 2 rhoptries docked to them. So, but who knows, maybe we don't know that once the whole thing disassembled upon secretion, whether or not those components are being recycled back together, become the same RSA and for docking the next AV, or there will be another Rosette formed by other set of proteins. So we don't know. Yeah.
Well, it's lots of very cool stuff and very cool, like, stuff that you figured out already and new questions to ask.
Yeah, it's super cool, super fun. Yeah, as a structural biology, this is really rewarding.
Yeah,
Yeah.
as somebody who doesn't know, oh, go ahead.
I was just to echo what Jason says. The graphics you have are excellent ways of portraying things. So, I know that making those kind of graphics can take a lot of work. So those were awesome.
Thank you. Hi, David.
Yeah, question from David Sibley. Go ahead.
Yes, this was really interesting. Some reason my video isn't working, but I really enjoyed your images. I wanted to ask about the position of the conoid during the imaging. So as you know, the conoid is protruded during invasion and retracted otherwise. And so what I'm wondering is, does the engagement of the rhoptry with the AV or the positioning of the AV, does it change when the conoid is out versus in? And is that part of the assembly process?
Yeah, so far we did not see clear difference in terms of, because we have tried to separate the tomogram into conoid protruded and retracted set, and then generate a subtomogram average of that part separately and compare their structure. At our resolution, we did not see clear difference. At our resolution and nanometer resolution. So far, we don't think at this resolution, there is some specific activity is related to conoid protrusion. But there might be some more subtle differences. Actually, it's related. We don't know. Yeah.
Okay, thank you.
Yeah, thank you, David.
Any other questions? I had one sort of a general question. Not knowing much about these organisms, what genetic tools do you have available to you to go in and make changes, mutations? Now that you have ideas about what proteins to target, like what do you have in hand? Could you go and make, target some of these to try and make these complexes so you can sort of freeze the infection events so that you can go and, because I think trying to find it by searching through tomograms is probably going to take a long time. Right. So
yeah. Definitely. So people like a parasitology experts like David, they have now the genetic tools to do genetic manipulations of this type of parasites. They were able to introduce taggings onto specific proteins, and then they can do induced knockdown. Because some of the components, the problem is that if you remove them from the beginning, then you will not be able to culture them because they no longer can invade host. So you cannot culture them. So one way people remove individual components is they put a tag on them and then just allow them to grow normally. But when you want to remove them, you induce the degradation of that protein with the tag by conjugating them with or connecting them with protein degradation machineries to chew away those things. And that can help us to start remove materials and then do differential comparisons between wild type and mutant tomograms. And that can be done. Yeah. So, and also if you can label the protein with a big tag, and if that will not affect the function too much, you can still culture them. We can also look for additional densities and compare with the wild type tomograms, and we might be able to pinpoint location of specific proteins. So all those can be tried. Yeah.
Yeah, lots of cool things to do. That's exciting. Great. Any other questions? If that's the end, Yi-Wei, thank you very much for a fascinating talk. Really great. Much appreciated. Thank you, everyone, for joining. I hear that Cryo-ET is, the methods development is going crazy. There's lots of very smart people producing lots of new software and new approaches. So the resolutions are just going to get better, and the amount of these sorts of questions that we can address, I'm looking forward to seeing them. So
thank you so much. Yeah, it definitely is complementary to all the other methods in the field. Definitely, people need to work together so we can have like different informations and to have fun together. This is really fun.
That's great. All right,
Awesome.
great.
Thank you so much, everybody.
Thank you.